Next up, video splicing from the keynote.
Well, sorry about the delay. I guess I'm the token splicer for the RNA Symposium. Every RNA Symposium has to have a RNA splicing session. Yeah, exactly. So I'm going to talk about a project that uses computational predictions. And just as a brief introduction, we all know that alternative processing is a very prevalent pathway in gene expression. More than 90% of all human genes are alternatively spliced. There are at least 2, on average, or 3 different mRNA isoforms generated from multi-mRNA genes. And the impact, as Yangshin already talked about for alternative APA, is very large and large. So typically, when we talk about alternative splicing, it comes in different flavors. For example, the one that most of you are familiar with is what we refer to as exon skipping. That's the complete deletion or inclusion of an exon. However, there's also alternative 5' and 3' splice sites. And essentially what this means is that the exon is contained, yet it's going to be a little bit longer on one side or on the other side. The impact of those is clear. You lose a protein domain, you may shift the reading frame, so the biological impact is relatively great. There are other types of alternative splicing that I'm not going to talk to you about in much detail, so I'm going to focus on these two. Just to give you a biological framework for what I really want to talk to you about is we have been interested in understanding whether alternative RNA processing, alternative splicing plays a role in breast cancer. And so you have a normal cell and you have a malignant cell. We know there's cancer progression, and we know from the literature there are alternative splicing changes that occur all over. So we had multiple questions. The first ones are really how much alternative splicing really associates with breast cancer formation. And the more interesting one to us, because we're really interested in figuring out what are the molecular mechanisms, why do we get alternative splicing, is for those changes that you see, what are the molecular causes for them? And the third one, that's not listed here, is what's the biological impact. That's what we count them up. Now, to do these kinds of experiments, as most of us do nowadays, we do transcriptome analysis. And as Yangshan, I'm a proponent for telling you that just looking at gene expression is not enough. You got to do a little bit more because, as I said, these mRNAs come in very different flavors and they may have very different functions. So we couple our RNA-seq analysis with an analysis that actually looks at the differences in local exon inclusion or local alternative splicing. The program we use is called MESO, and basically the idea is that you're looking for these exon-exon junction leads that either designate inclusion as this red and black one compared to the brown and black one that designates exclusion. And based on the number of leads that you observe for any alternative splicing event, you can determine the fraction included, you can compare condition A with condition B, you can figure out are there changes in alternative splicing. So when you do this with a bunch of breast cancer cell lines, you interrogate thousands of different possible alternative splicing events. I'm going to highlight here, these are the breast cancer cell lines that we evaluated. Some were positive, some were negative. And this is just a blow-up of this skipping event, the whole exon loss. And basically, of the thousands of events, of alternative splicing events that you do see in each of these cell lines, when you compare them to the normal control, hundreds display differential splicing. Okay, so this is a typical sort of RNA-seq experiment. You get hundreds of things. And you can do analyses like how many overlaps do we have, and you end up with, an enormous actually number of 76 that overlap, for example, between all of the cell lines that are negative. And what this means is that in addition of gene expression level changes as markers, you can use this information and actually establish novel breast cancer markers that are based on alternative splicing. So you can also use these when you start to do the go analysis terms, et cetera, et cetera. But that's where most of the RNA-seq experiments end. And what we really want to understand is, well, how do you get there? Okay? Is there some level of regulation? So the spliceosome obviously is in a different environment, and what are the molecular cues that are given to it to make those different decisions when you're in a normal breast cell or in a cancer cell? So let me talk a little bit about regulation of alternative splicing, and this is, many of you have seen this before, this is sort of a summary of years of experiments from many, many laboratories around the world. So basically what we're looking at is an internal exome, and what I've tried to do is sort of highlight the kinds of molecular cues that are given to the spliceosome so it makes a decision, yes, this region of nucleic acids is an exon or it is not. So obviously we have the splice sites. These are nucleotide sequences that designate the intron-exon junction. And they come in various flavors. They're not always the same sequence. And based on differences in the sequence, there are different efficiencies in how these splice sites are recognized. More importantly are these binding sites for RNA binding proteins that Rob just talked about. And they come also in different flavors, namely those proteins that have classically been described as, well, these are enhancers, so signaling take me as an exon. Or they are repressors, signaling leave me alone. I'm not an exon. And so we know throughout the years that we have 10, 20 SR or SR-like splicing enhancer proteins. There's a whole shopping list of them. Equally, we know of the splicing repressors. They're called H&RP proteins, so there are many, many of them. In addition to these proteins that we know interact and regulate splicing, and the way all of this was found out was by the hard kind of work where you look at one particular exon, and for years you study it and figure out, well, what binds there, what's the identity, and what does it do? So, in addition to these, we also know that RNA secondary structure is important. Makes sense. If you are involved in a stem loop structure, then obviously a protein can bind, as we have and the exon architecture. Last but not least is the action of making the RNA itself conform to. So, if we really want to understand the mechanisms, we should maybe think about whether we can predict splicing. Okay. And so, what we have started is a project to establish what is also referred to as a splicing code. The splicing code for us comes in actually two flavors. The first one we refer to as the general code. The general code asks the question, okay, if you are a region of nucleic acid, are you going to be an exon that's a cassette exon? Are you likely to be involved in alternative 5' selection? Are you likely to be involved in any other type of alternative splicing? What are the signals, if there are, that designate or pre-designate a region, an exon region, to be involved in one or multiple types of alternative splicing? The other one is really when we compare two biological settings, like normal and breast cancer. And what we do know is that, well, there are alternatively spliced exons, but they're differentially alternatively spliced in the two different conditions. And what are the molecular reasons? So, how do we go about setting up a splicing code? Basically, what we do is we go back to all these what I refer to RNA features. And let's say, take a splice site, for example. This splice site forms a base pair with a U1 snark. And based on the number of base pairs it can form, it's a strong splice site or it's not a strong splice site. Numerically, you can designate or you can calculate whether a splice site is strong or not. So you can designate numbers for all of these RNA features that I'm summarizing here. And in the end, we come up with our short shopping list of 260 different RNA features. This is a short shopping list that can be extended, and we have tried to extend it to 1,000, but it doesn't really improve what we do. So essentially, each exon has a combination of these different features that in cell type A designates the exon to be highly recognizable by the spliceosome, and in a different one it doesn't. And so what we do at a computational level is to calculate, describe each individual exon within the human genome with numbers, exon length, intron length, etc., 3' splice score, etc., 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 binding affinity to the recognizable SR proteins or Asian proteins. So we then take these features that we have derived for each of them, go into a source of known alternative splicing events. Those can be ESVs, they can be deep sequencing data, and out of those, we filter out all of the alternatively spliced, all-5s, constitutively spliced N-sets. And so we make these different groups. We evaluate their features, and when we toss each of these groups into a machine learning, I just call it a washing machine, that's probably the best way for me to describe it, and figure out, can we differentiate between these groups of alternative splicing? The way we evaluate the performance of that is wow.
Questions for Rob?
We carry out raw curve analyses. Rob already showed you one. Basically, what you plot is the rate of false positive versus the rate of true positive. And you're happy if you get a curve like that we expect. So the larger the area under the curve, the better you are doing. And if you are in the diagonal, basically your experiment failed. So before we started doing these analyses, these machine learning analyses, we also realized that our levels of inclusion of these thousands of different alternatively spliced exons, they come in these sort of binomial fashions. And that is that many are not highly included, very many are very highly included. And this is true for all of it, except the all-3s and the all-5s. So what we decided to do was to not include or separate out the highly included ones because they're more likely behaving like constitutive exons and compare them to the less included ones. So just to give you an idea of our performance, when we compare constitutive exons with highly included ones, the experiment fails. We cannot differentiate without these RNA features. There's no way for us to differentiate between a highly included exon. However, when we look at these lower inclusion values, we get a phenomenal differentiation between these two classes. You can carry this one out further for comparison between alternatively spliced 3, alternative 5, all 3 versus all 5. And in all of the cases, you see that we do very well for the low inclusion, so more than half of all of them. However, we are not doing that well when we're comparing those that appear to be highly constitutive. Now, that's great. So you can use now these derived splicing codes to figure out, well, what in the end are these RNA features that differentiate an exon that's alternatively skipped to an exon that's alternatively 3' spliced. And so basically, what we want to figure out is which ones of these 260 features are the ones that give us the most information. or in other words, those that are trying that type of alternative splicing. So I'm plotting here. This is a little complex, but basically all features that associate with the 3' splice site are included. And when you look at the most highly ranked features, everything is 3' splice site. Right? So this means the 3' splice sites form, whether there's an alternative 3' splice site upstream. We also have a conservation feature. What is lacking here are any binding sites for regulatory proteins, these enhancers, these repressors. They never show up. They don't show up. So the designation of whether you as an exon are going to be alternatively spliced one way or another is not influenced by the enhancers and repressors. It's really influenced by your immediate sequence contents. Now you see similar things when you look at the alternative skip exons. And here what you see are an overrepresentation of 5' features and 3' features. Okay, so that's sort of the general splicing code. So if we can use these features, we can use machine learning approaches, we can predict what kind of alternative splicing an exon is going to be playing a role. And so the most important features are really the splice site scores, but also conservation. Conservation for us is a cheat feature because it tells us whether there is a region that is potentially important and therefore conserved within evolution. There might be structure features within that, there might be sequence features that bind to proteins we don't know. There's absolutely no strong contribution of the classical splicing regulators when it comes to this designating an exon as an alternative skip or alternative follow. We have very good performance when we consider the low and mid range of alternative splicing, and our feature analysis fails when it comes to high range, which basically supports the notion that we had in the beginning that these highly included exons, they behave more or less like constitutive exons. Okay, so now let's move to the second predictor, and that is you have two conditions: normal breast cancer. We identified hundreds of different exons that are alternatively spliced. Can we take these pools of exons and figure out whether there are some molecular features that may give us hints about what the mechanism is? So essentially, what we're going to do is we're looking at an exon that's alternatively spliced in one condition and significantly alternatively spliced in a comparable condition. So basically what we're doing then is we're taking the exons that appear to change when you compare normal and breast cancer. We feed them in the splicing code, and out comes the report of what molecular features are important. And that is going to be my last summary slide. Basically, what you said, rather than showing you raw curves, I want to summarize what pops up then. When you compare biological conditions 1 and 2, normal versus breast cancer, what you now see is multiple splicing regulatory protein binding sites pop up, and it's providing sufficient information content to make a differentiation. So in other words, for those exons that demonstrate increased execution, the SR protein 1 and SR protein 2 pop up as important in terms of regulation. So what this means now is that I have a roadmap to set my students conclusions onto trying to figure out whether alternative splicing plays a significant role in cancer progression. And what the roadmap basically tells us is that these SR proteins and the FOX proteins here are important in that transition or in the generation of the alternatively spliced exons that we use in breast cancer. And so to summarize then, the second part is basically we can use the splicing code to show differences in alternative splicing. This is really, the second part is a really much harder problem to solve. Our raw curves are not as great as I've shown you in the earlier part. We're more in the areas of, I believe, 70 to 80% under the curve. But when you do compare different biological systems or conditions, that's when you see regulators fall. So overall conclusions are then that the type of alternative splicing that an exon undergoes is dictated by its immediate sequence context. Whether or not you're going to be more or less alternatively spliced depends on the presence and activity of splicing regulators that are going to be differentially expressed as in breast cancer, for example, and so with that, I'd like to end and thank people who did most of the work. That's Dr. Bush, who's unfortunately no longer with us, and Gary Klemens, who did a lot of economics. So with that, thank you very much.